Abnormal methylation of tumour suppressor genes. It is known that abnormal DNA methylation is common in the development of a variety of tumours. The most common abnormality is hypermethylation, which means more methylation. The process by which hypermethylation may lead to cancer is as follows. Hypermethylation occurs in a specific region, the promoter region, of a tumour suppressor gene. This leads to the tumour suppressor gene becoming inactivated. As a result, transcription of the promoter regions of the tumour suppressor gene is inhibited. The tumour suppressor gene is therefore silenced. As the tumour suppressor gene normally slows the rate of cell division, its activation leads to increased cell division and formation of a tumour. Abnormal methylation of this type is thought to occur in the tumour suppressor gene known as BRCA1 and leads to the development of breast cancer. Another form of abnormal methylation is hypomethylation, which is reduced methylation, and this has been found to occur in oncogenes, where it leads to their activation and hence the formation of tumours. Estrogen plays a central role in regulating menstrual cycle in women. It is known that after the menopause, a woman's risk of developing breast cancer increases. This is thought to be due to increased estrogen concentrations. At first, this seemed paradoxical because the production of estrogens from the ovaries diminishes after menopause. However, the fat cells of the breasts tend to produce more estrogens after the menopause. These locally produced estrogens appear to trigger breast cancer in postmenopausal women. Once a tumour is developed, it further increases estrogen concentration, which therefore leads to increased development of the tumour. It also appears that white blood cells that are drawn to the tumour increase estrogen production. This leads to even greater development of the tumour. How then can estrogen cause a tumour to develop? Basically, the estrogen effectively activates a gene by releasing an inhibitor molecule that prevents transcription. If the gene that estrogen acts on is one that controls cell division and growth, then it will be activated, and its continued cell division could produce a tumour. It is known, for example, that estrogen causes proto-oncogenes of cells in breast tissue to develop into oncogenes, and that leads to breast cancer. Risk factors in cancer. Smoking. Not only smokers are in danger, those who passively breathe in tobacco smoke also have high risk. Remember, benzopyrene being a mutagenic agent. Diet. What we eat and drink affects our risk of contracting cancer. Strong evidence that low-fat, high-fibre diet rich in fruit and vegetables reduces risk. Obesity. Being overweight increases risk of cancer. Physical activity. People who take regular exercise are at lower risk of some cancers than those who take no exercise. Sunlight. The more someone is exposed to sunlight or light from sunbeds, the greater the risk of skin cancer. Epidemiologists collect data on diseases and then look for correlations between these diseases and various factors in the lives of those who have them. The world's longest-running survey of smoking began in the UK in 1951. This survey, and others elsewhere in the world, has revealed a number of statistically significant correlations. For example, a regular smoker is 3 times more likely to die prematurely than a non-smoker. The more cigarettes you smoke a day, the earlier the average smoker dies. Smokers who give up the habit improve their life expectancy compared with those who don't and continue to smoke. Long-term smokers are more likely to die early as a result of smoking. Incidence of pulmonary disease increases with numbers of cigarettes smoked per day. Smokers make up 98% of emphysema sufferers. Epidemiological statistics show correlations between lung cancer and smoking. A man smoking 25 cigarettes a day is 25 times more likely to die of lung cancer than a non-smoker. The longer a person smokes, the greater the risk of developing lung cancer. When a person stops smoking, the risk of developing lung cancer disease decreases and approaches that of a non-smoker after around 10 to 15 years. The death rate from lung cancer is 18 times greater in a smoker than a non-smoker. Experimental evidence linking smoking to diseases. Scientists carried out experiments in the 1960s in which dogs were made to inhale cigarette smoke. The smoke was inhaled, and those dogs that inhaled it developed early signs of lung cancer. Scientists then carried out a further series of experiments that allowed them to formulate a new hypothesis. Machines were used to simulate the action of smoking and to collect the harmful constituents that accumulated in the filters. These were then analysed chemically and each constituent was tested in the lab for its ability to damage epithelial cells and mutate the genes they contain. This was done by adding tar to the skin of mice or to cells that had been grown in culture. As a result of such tests, it was shown that the tar found in cigarette smoke contained carcinogens. The constituent chemicals of the tar were each tested and 1-benzopyrene was shown to mutate DNA. The scientists still had to demonstrate precisely how it caused cancer. They carried out experiments, and these showed that benzopyrene is absorbed by epithelial cells and converted into a derivative. This binds with a gene and mutates it. Another experiment showed that this mutation led to uncontrolled cell division of the cells. Even this was not proof. In further experiments, scientists showed that the mutations of the gene in a cancer cell occurred at 3 specific points on the DNA. When the derivative of benzopyrene from tobacco smoke was used to mutate the gene, it caused changes to the DNA at precisely the same points. The evidence was now conclusive. Smoking tobacco could cause lung cancer. This is not to say that it always does, but simply that there is an increased risk. It's always about probabilities and not certainties.